Okay, sports fans, here we go. We're moving on to the kind of the second part of the kind of search for DNA here. So the first part was, hey, what molecule is actually transferring the genetic information? And thanks to four scientists, we figured that out. Who are those four scientists? That you're supposed to be saying it. Okay, in case you forgot, it's Frederick Griffith, and he figured out that some sort of genetic material was being transferred from dead bacteria to live bacteria, and that was called transformation. Then Avery figured out that the molecule that did transformation was DNA, because when he took DNA away, transformation stopped happening. But when he took protein away, or RNA, or lipids, or carbohydrates away, transformation still happened. So he knew it was DNA. Hershey and Chase backed up Avery's experiment by putting radioactive markers in the coat and in the DNA of viruses, and then looked to see where did that radioactive material end up. Did it end up inside the cell or outside the cell? Turned out that the DNA marker, which was phosphorus, ended up in the molecule inside the cell. So they realized, hey, DNA actually is the genetic information. It's that molecule. So then once they figured out, once everybody accepted, okay, Hershey and Chase and Avery figured this out, once they accepted that, they started going, well, what exactly is the structure of DNA? Everybody knew DNA existed. They didn't know how it was structured. They didn't know how it worked. They didn't know how it did what it did. And that's where the focus of the research began to happen. And all of this stuff started happening around the same time, kind of early 50s. Okay, so today we're going to talk about a man named Erwin Chargaff. And Chargaff, he was the one that kind of figured out the first part. And it was probably 1950, 51, I think, maybe 52. But what Mr. Chargaff, this dapper-looking fellow, was trying to do is he was trying to see kind of ratios. He was studying organisms to see what their ratios of nitrogenous bases were in their DNA. Now I'm going to explain what nitrogenous bases were, okay? So, well, if I grab them, there we go. So here are the nitrogenous bases. Let's see if I have room for it. I think I'm going to make it a little smaller here. There we go. Here's our nitrogenous bases, and we have 5 of them. We have adenine, and at some point I'm going to want you to be more familiar, but not yet. This is adenine, and we represent that with an A. This is guanine. We represent that with a G. This is cytosine. Guess what? Yeah, that's right, a C. This is thymine, a T, and in the middle is uracil. Well, it turns out uracil is not in DNA. It is only in RNA. So we'll talk about uracil later, but for right now, understand that uracil does not exist in DNA. That is one major difference between DNA and RNA, is that uracil exists in RNA, and in DNA we have thymine. So thymine is not in RNA, it's only in DNA. Uracil is only in RNA, not in DNA. And the other big difference is DNA is a double helix. It has 2 strands that wrap around each other, whereas RNA is a single helix. It's one strand that spirals around, and that's kind of how it's set up. Those are the 2 big differences. I guess the last one would be that DNA, the sugar in DNA is called deoxyribose, and in RNA is just called ribose. So there are 3 differences for you to pay attention to between RNA and DNA. Okay. Okay, so what he was curious was in each animal, how much adenine is there, how much guanine is there, how much cytosine, how much thymine compared to each other. And that's what he was really focusing his research on, okay? So let me show you what he figured out. Let's move those big guys away. Really, it's very simple. He figured out when he looked at organisms that he would count, like he did some chemical tests and determined sort of a weight, the weight of the adenine. He wanted to figure that out, and then so he would take DNA, and in every animal he'd break that DNA apart, or every organism, and he would see how much adenine was there, how much guanine, how much cytosine, how much thymine. And as he paid attention, he started to notice something cool. There was always the same amount of adenine as there is thymine. That amount was always the same, okay? And it turns out also that the amount of, whoa, that's not what I wanted to do, that the amount of guanine was always the same as the amount of cytosine. Turns out they were always equal, and he was kind of perplexed by that. It especially became interesting because it didn't turn out that the amount of adenine equaled the amount of cytosine. That did not work. It never worked. The amount of adenine was never the same as the amount of cytosine. And the amount of guanine was never the same as the amount of thymine. Okay? This is a big deal. And I know it seems really simple, but at the time nobody knew that. Okay? So, he found out that basically A always equaled T, G always equaled C. These are the rules. These are Chargaff's rules. Adenine always equals thymine. Guanine always equals cytosine. But adenine never equals cytosine. Thymine doesn't equal cytosine. Adenine doesn't equal guanine. Thymine doesn't equal that. That's what he figured out. Okay, what's really cool is he looked at a lot of different organisms to see if this was true. He looked in animals, of course. It was true. So, check on that one. Check. He looked on plants. Was it true? Check. He looked on yeast. So he looked at fungi, right? Our old friend. Check. And even in bacteria. Right? Here's bacteria. Found it. The DNA in all of these always follow these rules. And what are those rules? First rule, adenine always equals thymine. That's rule 1. Rule 2, guanine always equals cytosine. And you could say cytosine always equals guanine, same thing. But the third rule, this does not equal this does not equal this. Okay? So this equals this equals this does not equal this or this. Those are the 3 rules. Now you understand it. Seems very simple, and you know what? It is very simple. The deal is, though, it became kind of one of the basic things they used for research on... Okay, sports fans, so there it is. What's same? These 2 are the same. What's different? These 2 are always different. Okay, what's it good for? All these organisms. Who did it? Chargaff. Hi. Okay, you should understand it all now. These are Chargaff's rules. If you don't understand it, talk to me or look on page 345 of your textbook. There's a little bit about Mr. Chargaff. Interesting, dapper fellow. Okay, if you don't have any questions, I'll see you tomorrow. If you do, please talk to me. All right, peace out, homies.